not that much of like aromatase, but it's detected almost the neuronal cells, not in the radial cells of the ventricle. And POA also we detect here, like neuronal cells. We also detect the signal in the midbrain area, tectum, and some part of the valvula. So we measure the sex steroids in the brain. We detect the high significant effect of estradiol during gonadal sex differentiation around 120 days period. And in the hypothalamus, it's increased in 150 days. But come to testosterone, it's almost significantly decreased from 110 to 150, not so much like estradiol. So we also compare male and female brain to study about important steroidogenic enzyme genes during the development. So we detect the intensity of the band at 110 in the forebrain. In the midbrain also in 110 for CYP11A1 and keepvid HST and CYP17A1. And in hypothalamus we detect for CYP11A1 and CYP17. In black coffee we detect significantly increase in 110 days. So the summary, all the steroidogenic enzyme transcript increased over 110 days in midbrain and decreased on 150 days in midbrain for CYP11A1 and CYP17A1 in hindbrain for 3-beta HST. And CYP19A1B mRNA expression had gradually increased from 90 to 150 in forebrain and hypothalamus. However, a significant high expression was found at 110 in forebrain and 130 and 150 in hypothalamus. The brain aromatase activity and estradiol, not testosterone, were apparently increased at 110 and 120. So the RT-PCR demonstrating that the steroidogenic enzyme genes, especially brain aromatase, has similarly expressed during gonadal sex differentiation in female and male fish. And the CYP19A1B transcript localized on the radial glial cells along the telencephalonic ventricle, preoptic area, and diencephalonic ventricle. However, the HST3P1 has not been overlapping with that of CYP19A1B because we detected in the neuronal cells. And the second experiment we did the in vivo effect of E2 and the fish to detect the transcript of aromatase and pCNA. So we conducted the experiment, one is the control group. We injected day 1 the coconut oil and day 5 coconut oil. And for experimental group, we injected E2 the day 1 and day 5. And day 6 we collected the sample, forebrain, midbrain and hypothalamus. So see the result. The CYP19A1B significantly increased when compared to the control, especially in the forebrain. And in the hypothalamus also it's increased. But compared to hypothalamus and forebrain, the forebrain is more significantly increased. Oh, sorry. In the pCNA, it's almost increased in the E2 injected group, but compared to the control, that much not significant different. So this is the distribution of aromatase immunoreactivity cells we compared in the control and E2 group. You see the E2 injected group, we detected aromatase immunoreactivity cells is very strong and the radial glial cells of the telencephalonic ventricle. This is the magnified picture. Here you can clearly see the radial glial cell projection from the telencephalonic ventricle here. And the POA area also, we detected the aromatase immunoreactivity cells. So the summary, exogenous E2 stimulated CYP19A1B transcript in forebrain and hypothalamus, but not in the midbrain. However, no significant effect on pCNA transcript in forebrain, midbrain and hypothalamus. Aromatase immunoreactivity was on the radial glial cells of the telencephalonic ventricle, preoptic area and lateral recess. And further, the aromatase immunoreactivity was more high in E2 injected group compared to the control group. So we concluding all the steroidogenic enzyme genes are highly expressed during the brain development during the gonadal sex differentiation. And CYP19A1B expressed especially on the radial glial cell. However, the 3-beta HST signals seems to correspond to other cell types such as neuron. The exogenous E2 upregulated CYP19A1B in differentiated brain. So finally we demonstrated for the first time to our knowledge that differentiation or further development in the brain during gonadal sex differentiation is not a gender related circumstance. Thank you very much for your kind attention.
Any question?
Due to the time time is quite limited right now, so we thanks for this talk and leadership discussion. Our next speaker, and is going to be the last talk, is from Dr. Chen's lab again, and Adimulam Luna. And today's topic is long-term salinity effects on stress genes in tilapia.
Good evening to everyone. Today I'm going to talk about the stress genes. Stress linkage, that is long-term salinity effects on the stress genes in bosonic tilapia. That is during seawater acclimation, how these fishes going to face the seawater challenge and the involvement of neuroendocrine hormones during stress response. Further, we choose the tilapia for this study to study about the stress response. The stress response, actually the tilapia is generally urichalentilias. It can tolerate a wide range of salinity, but this seawater tolerance is limited to some extent. And however, in the stress response due to a stressor is the essential phenomena. In this regard, the stress response is divided into characteristics into 3 types based on depending upon their response. That is, one is behavioral, physiological, and cellular stress response. The involvement of neuroendocrine hormones come under the category physiological stress response, especially in the primary stress response. So during stress, the hypothalamic pituitary interrenal axis are activated by the neural peptide CRH from the hypothalamus. This hormone stimulates the anterior pituitary to secrete another hormone, ACTH, adrenocorticotropic hormone. And finally, this hormone stimulates the internal tissues to secrete the stress hormone cortisol. This cortisol finally binds to the cortisol receptors in the target tissue to maintain a homeostasis in teleas. So, the recent study about the cortisol receptor in higher vertebrates, maybe in such as mammals, the mineralocorticoids and glucocorticoids exert their functions by their specific receptors, glucocorticoid and mineralocorticoid receptor. But in teleas, due to the lack of mineralocorticoids, the cortisol carry both glucocorticoid and mineralocorticoid function by these receptors to maintain the homeostasis. So one of study in rainbow trout, they reported the MR has a similar affinity with cortisol as in GR. The another study also reported the binding capacity of cortisol to MR was 10-fold higher than that in GR in teleas. So far in teleas, 2 GRs, that is 1 and 2, and 1 MR were found, and these cortisol receptors are widely expressed in brain and gill of Mozambique tilapia. So, presence of these multiple cortisol receptors may the complicated glucocorticoid signaling during stress response. So, in this study, we hypothesis during seawater acclimation, especially in the long-term seawater acclimation, both brain and gill need their support to maintain the stress response, to maintain the homeostasis. So further, we conduct the experiment. We transfer the some of the fish from freshwater to seawater and kept for 30 days. And then after 30 days, we sampled brain, pituitary, gill, and brain for quantify the stress hormones and their receptor transcripts, and localization of some of the important cortisol receptors in the gill, and also measure the osmolality cortisol level in plasma to monitor the stress response. So from this result experiment, we obtained the increased level of osmolality and cortisol in the plasma levels. This means during the long-term seawater acclimation, the fish increase their cortisol level and osmolality level to improve their seawater tolerance, and also maintain their iron ion balance according to the between the two gradients. And we also measure the mRNA transcripts of in brain. So in this, during seawater acclimation, the stress hormones CRH and CRH receptor and cortisol receptor, one of the cortisol receptor, GR1, is significantly expressed in forebrain, hypothalamus. These three, and whereas in GR2, another hormone GR2 and MR were significantly increased in all parts of the brain, forebrain, midbrain, hypothalamus. The similar expression in the brain region. And coming to the pituitary, the significant continuous exposure of the stress hormone from the hypothalamus, that is CRH, it desensitizes the pituitary corticotrophs cells. So it downregulates the